Hey, welcome to The Explainer. Today we're diving deep into a field that is literally changing the world as we know it, biotechnology. I mean, think about it. This is the science of reaching into the very instruction manual of life, DNA, and making edits. We're talking about hacking the code of life itself. So let's get into it.
You know, in a way, biotechnology has been around for ages. For thousands of years, we've been using what you might call traditional biotech, making curd, bread, wine, vinegar. These are all processes that use living organisms. But what we're seeing today is something completely different. Modern biotechnology, which is built on recombined DNA technology, isn't just about using what nature gave us. It's about programming a living cell, fundamentally rewriting its genetic code to do things it would never do on its own. We've basically gone from the kitchen to the cutting edge of science.
So how did we make that incredible leap? How do you go from fermenting grapes to literally programming a cell? Well, it all hinges on one revolutionary concept that changed everything.
And that concept is genetic engineering. Now, the key words here say it all. Manipulation, desired gene, predetermined way. This isn't about luck or chance. It's about scientists choosing a specific trait they want and then deliberately, precisely, engineering it into an organism's DNA. And it happens in vitro, which just means in a lab, in a test tube, outside of the cell itself. It's total control.
All right, so to do this kind of microscopic surgery, you can't just use any old scalpel. You need a highly specialized set of tools. So let's imagine we're opening up the genetic engineer's toolkit and see what's inside.
First up, and you could argue these are the most important tool in the kit, are restriction enzymes. The perfect way to think of these is as molecular scissors. But they're not just any scissors. They are incredibly precise. Each enzyme is programmed to find a very specific sequence of DNA letters and cut right there. That incredible precision is the key that unlocks the whole field of genetic engineering.
And these scissors even come in different models, if you will. You've got endonucleases, which make a cut from within a strand of DNA, kind of like taking a single link out of the middle of a bicycle chain. And then you have exonucleases, which snip off the letters from the very ends of the chain. Having both gives scientists amazing control over how they isolate the exact piece of code they need.
Okay, so you've used your molecular scissors and you've cut out the gene you want. Great. Now what? What do you do with it? How do you get it into the new organism? For that, you need a delivery vehicle. And in biotech, we call this a cloning vector. It's typically a tiny loop of DNA, often a plasmid borrowed from a bacterium, that acts like a microscopic truck to carry your gene of interest into a host cell. Our third tool is a real game changer, and it solves a big problem. You usually start with a ridiculously small amount of DNA, maybe just 1 or 2 copies. That's where PCR, the polymerase chain reaction, comes in. This technique, developed by Kary Mullis, is basically a genetic photocopier. It can take one tiny segment of DNA and amplify it into millions or even billions of identical copies in just a couple of hours.
So how does this genetic photocopier actually work? It's really just a simple 3-step cycle that you repeat over and over. First, denaturation. You heat up the DNA, which causes its 2 strands to unzip and separate. Second, annealing. You cool it down just enough for these little DNA primers to lock onto the strands. And third, elongation. An enzyme comes in and builds a brand new matching strand. You do that cycle, say 30 times, and boom, you've gone from 1 copy of DNA to over 1 billion. It's incredible.
Okay, we've got our toolkit. We have our scissors, our delivery truck, and our photocopier. Now let's put it all together and walk through the actual step-by-step process for creating a brand new genetically modified organism.
When you boil it all down, the entire process is really just 4 main steps. First, you've got to isolate the gene you're actually interested in, say the gene that makes a firefly glow. Second, you use your tools to insert that gene into your vector, your delivery truck. Third, you transfer that vector into your host organism, maybe a bacterium or a plant cell. And finally, and this part's really important, you have to select the cells that actually accepted the new gene and then let them multiply.
This diagram shows the whole thing beautifully. You start with your plasmid, that's the vector from an E. coli bacterium, and your DNA of interest. The molecular scissors cut both of them. Then, another enzyme acts like a biological glue to paste them together. What you get is called recombinant DNA, or rDNA.
You then introduce this new, modified plasmid back into a host cell. Now, every time that host cell divides, it not only copies its own DNA, it also copies your new gene, creating these transformed cells that can now produce a whole new protein. So we've covered the how, now for the really fun part, the what. What have we actually managed to create with this amazing toolkit? I'll tell you, the applications are already completely rewriting our world. This right here is one of the most famous and frankly coolest proofs of concept ever. Scientists literally took the gene for luciferase, that's the enzyme that makes fireflies light up, and they put it into a tobacco plant. And what happened? The plant glowed. It produced its own light. It's just this stunning visual proof that you can take a gene from one kind of organism and make it work in a completely different one. But it goes way beyond just cool experiments. Look at the impact on agriculture. Through a combination of genetic modification and advanced breeding, milk production has skyrocketed. A cow used to produce maybe 2,000 to 3,000 liters a year. Now it's closer to 8 or 10,000. Or look at hens. Egg production has shot up from around 70 eggs a year to over 250. These are just massive leaps in productivity.
And now we're even turning plants into tiny biological factories. It's a field called biofarming, and some of this stuff sounds like pure science fiction. We're talking about tobacco plants that produce human growth hormone, plants that create antibodies against HIV. We can even get proteins from plants that act as a surgical superglue, or engineer them to produce artemisinin, which is a critical anti-malarial drug. It's wild.
And of course there's medicine, which might be the ultimate frontier. The goal here is gene therapy. The idea is that for genetic diseases like sickle cell anemia or hemophilia, we don't just treat the symptoms anymore. We go right to the source, the DNA itself, and we fix the typo. We replace or alter the single faulty gene that causes the whole problem.
But, with this kind of incredible power comes enormous responsibility. This technology forces us to stop and ask some really big, really difficult questions about safety, ethics, and who's in control.
Yeah, every new technology has trade-offs. Let's take herbicide resistance. On the one hand, the benefit is pretty clear. Farmers can spray their fields for weeds without worrying about killing their crops. It's more efficient. But on the other hand, there can be unintended consequences. We've seen, for example, that monarch butterfly populations have declined, and that's been linked to the loss of their food source, milkweed, which is often killed off by the herbicides used on these resistant crops.
And then there are potential health concerns. There's a famous case where a gene from Brazil nuts was put into soybeans to make them more nutritious. Well, they had to scrap the whole project because they found out that people with Brazil nut allergies were also allergic to these new soybeans. And beyond specific cases like that, there's a bigger concern. This is all still pretty new technology, so we just don't have decades of data on the long-term health effects yet.
Finally, you get into this really murky area called biopiracy. This is essentially when a company patents a natural resource or a piece of traditional knowledge without giving credit or compensation to the people who discovered it. For example, a U.S. company patented a method for using the neem tree as a pesticide, a method that had been known and used in India for centuries. Same thing happened with basmati rice, where an American company patented a rice line they developed from traditional Indian strains. It raises these huge questions about who gets to own nature. And that's where we are today. We have this toolkit that can do incredible things: cure disease, feed the world, solve massive environmental challenges. But it's a toolkit that also comes with very real risks and deep ethical questions we have to wrestle with. We've gained the power to fundamentally reshape our planet. So the ultimate question, really, the one that's left for all of us to answer is, what should we choose to build?